Without a doubt, my favorite kingdom of life is the eukarya. Typically, they have more complex and robust cellular systems compared to their prokaryotic relatives, and have many quirks that make them incredibly diverse and unique. While some eukaryotes hog all of the press and media coverage, like humans or agricultural crops, I find the lesser sung tales of the microscopic eukarya to be most interesting. The kingdom of fungi lies right at the intersection of plants and animals, where their cellular shape and processes are quite similar to both. They can live as multicellular molds and form sexual structures in the shape of macroscopic invisible mushrooms, or they can live as unicellular organisms in a yeast form that live in a community of relatives known as a colony. Some fungal species are able to switch between these forms, while others are found only as one form, or as a combination of both. While unicellular yeasts are microscopic and primarily reproduce via budding from one cell into two, molds present as long filaments of multiple cells and can reproduce through spore production and dispersal. While fungi of either form can be found in many different niches in the environment, molds can play a very interesting ecological role when they are found in soil.
In the environment, a very interesting interaction can occur between fungi and plants. Fungi live in the same environment as plants by reaching down into the soil to reach nutrients that the plants would otherwise not be able to reach. Interestingly, the plants and fungus can form a symbiotic relationship in which the fungus provides nutrients like phosphorus and water that are farther than the plant's roots can reach, and can give these nutrients to the plant in exchange for nutrients like sugars and carbohydrates.
Filamentous fungi that have the ability to form this relationship with plants can form these relationships with many plants in the same area, allowing for the development of very complex mycelial networks. In fact, these networks can become so complex and widespread that they can cover an entire forest, taking up nutrients from the soil, digesting dead and decaying plants, and dispersing these nutrients to the plants that give sugar or other nutrients the fungus needs in return. The largest known organism to ever exist is not a blue whale or a prehistoric dinosaur, but rather a network of the saprophytic fungal species Armillaria ostoyae. Individuals of this species from a single forest have been measured to have an upper range of 50 acres to an incredible 2,385 acres in size. The largest individual that covers over 2,000 acres of space is thousands of years old, and is estimated to weigh tens of thousands of tons while continuing to grow at a rate of several feet per year, located in the Malheur National Forest in Oregon. While this is a fascinating organism, it is incredibly pathogenic and has only grown so large by killing trees and using its nutrients to increase its size. Instead of forming symbiotic relationships with plants like other mycorrhizal fungi, these organisms completely take advantage of the nutrients the plant produces and provides nothing but death in return. Not all soil fungi are completely evil, however. These symbiosis through transporting nutrients and fighting off pathogens that some fungi provide for a plant is incredibly ecologically important and fascinating. Even more, the diversity of life found in soil can be incredibly complex, where many kinds of bacteria, fungi, and other organisms can all be fighting for the same nutrients. Some fungi have developed an ecological role of mediating the interactions between different organisms found in the soil. Fungi of the Glomeromycota phylum have the ability to form endosymbiotic relationships with many different types of plants and even host their own intracellular bacteria. For a long time, it was unknown whether the endobacteria were true symbiotes and were living intracellularly, or if it was a byproduct of an endosymbiotic event, like the acquisition of ancestral mitochondria, where the endosymbiont is not actually alive. It wasn't until 1996 when a research group was able to conclusively determine that the endobacteria of one species of mycorrhizal fungi, Gigaspora margarita, was very closely related to species of the Burkholderia bacterial genera, and were definitely living inside of the fungal cell. To do this, the researchers used a 16S rRNA PCR sequencing technique to show that the endobacteria had a similar genetic makeup to the Burkholderia genera, and used a viability kit to stain living bacterial cells, which showed that these bacteria truly were replicating and living intracellularly. A few years later, a separate study looked at different species of mycorrhizal fungi and found that they housed similarly living endobacteria that were all genetically related to either the Burkholderia genera or the Mycoplasma genera of bacteria. Apart from these studies, the direct mechanisms by which mycorrhizal fungi evolved a symbiosis not only with plants, but also with an endosymbiotic bacteria have remained largely unknown. From all of the mycorrhizal fungi looked at, it appears as though endobacteria are mostly specific to the glomeromycota. In fact, this symbiosis has evolved to a point where both organisms require the presence of the other to grow. When Gigaspora margarita is cured of its Burkholderia-related endosymbiont, it exhibits a difference in morphology and hyphal production is inhibited. Additionally, these endobacteria are completely unable to grow on their own, which is a major contributor as to why it was so difficult to speciate them until the advent of sequencing. As the field of genomics has progressed, more and more studies have been able to look into the mechanisms of these symbiotic relationships. One such study looked at the genetic makeup of both the mycorrhizal fungi Diversispora epigeiae and its endosymbiotic bacteria, which are more related to the Mycoplasma species than to Burkholderia. They found that regardless of the genetic relatedness of the fungi, the endobacteria separate into two distinct lineages that are conserved across the glomeromycota. Compared to other species of fungi, Diversispora epigeiae was found to have lost genes involved in degradation of the plant cell wall, such as exocellulases and pectin-degrading enzymes, which would prevent the fungus from digesting its symbiotic plant. However, several key hemicellulose-degrading enzymes were found in the genome that are hypothesized to be involved in the process of the fungus colonizing the roots of the plants. While Diversispora epigeiae has a large proteome encoded by over 28,000 protein-coding genes, their endosymbiotic bacteria have a massively limited metabolism, with only around 600 proteins that they encode. Interestingly, the fungus is missing several key proteins involved in normal fungal metabolism, including its ability to generate crucial biomolecules like thiamine and certain crucial fatty acids. Similarly, the endobacteria show severely reduced metabolic capacity, having lost almost all genes required for transport and processing of lipids, carbohydrates, and amino acids. Thus, the fungus relies on nutrients provided by the plant to fill the role of these missing genes, while the bacteria relies on materials produced by the fungus. Interestingly, while the endobacterial genome is missing many of its expected metabolic genes, it contains several genes that are key for fungal metabolism. While these do not make up for any of the genes that is missing in the fungus, their presence and function could be beneficial for the fungus and could be an evolutionary reason why the endobacteria are tolerated by the fungus. This is a really fascinating exploration into the genetic makeup of organisms that rely on symbiosis and certain mechanisms that influence these relationships. Each individual in this environment, the fungus, the bacteria, and the plant, are all benefiting from the presence of each other. In return for sugars, a niche to colonize, and fatty acids that are missing from its metabolic pathways, the fungus provides nutrients through its mycelial network that the plant would otherwise not be able to reach. The endobacteria provides metabolic aid to the fungus while relying on the metabolism of both the plant and fungus due to its extremely limited genome. While the plant would be able to grow just fine in the absence of both, the fungus and its endosymbiont require symbiosis with the plant to provide necessary nutrients, in return allowing the plant to grow better than it could without such a relationship. Understanding and continuing to study symbiotic relationships between plants and the other organisms in their environment has a direct impact on human health through its agricultural significance. Promoting the growth of, or even adding mycorrhizal fungi to the soil of crops has some very important benefits. Not only do these fungi increase water and nutrient uptake by the plant, but they also protect the plant against a variety of stressful environments that are getting more and more severe across the globe. In many areas, drought, or the shortage of water, is an incredibly important concern that greatly inhibits the growth of plants and crops, which could lead to famine. The symbiotic relationship with fungi has been shown to greatly increase plant tolerance to drought through increasing water availability and promoting the plant to increase its water retention. Similarly, soils affected by large amounts of salt has been an increasing global problem as high salinity stress suppresses plant growth and yield. The presence of mycorrhizal fungi has also been shown to reduce this form of stress as well, where the fungi work to rearrange the salinity gradient to promote plant growth. Additionally, soils with heavy metal contamination like cadmium, nickel, or zinc not only inhibit plant growth and can even result in plant death, but are the cause of important human health concerns. The inclusion of mycorrhizal fungi in soil results in the movement of these metals away from the plant, where they are not hazardous for either the plant or the human eating the plant. Under stressful temperature conditions, either dangerously hot or dangerously cold, plant yield and growth is significantly impacted. The presence of mycorrhizal fungi helps the plant grow under both stresses by retaining moisture, secondary metabolites, proteins, and other nutrients under the ground where they would otherwise be lost by the plant due to these conditions. These fungi also protect against pathogenic microbes, including other fungi like the humongous fungus of Oregon, Armillaria astoye, that would rather kill the plant and use its resources for its own benefit. Fungi that live in the soil have direct and important impacts on human health through promoting plant survival in the dangerous and stressful conditions that are becoming increasingly widespread and concerning due to both climate change and poor agricultural or industrial practices. With the help of our fungal relatives, humans might even be able to survive in the horrible environmental future that we are continuing to make for ourselves.